Chapter 25, Mycoplasmas and Cell Wall Defective Bacteria.
The very dense chapter.
Extremely dense. But our mission today is to explain every single line of this text for you.
We are going to unpack everything.
Yeah, from their strange fried egg appearance in the lab to why your doctor might prescribe a very specific antibiotic and definitely not penicillin if you have a lingering cough.
And it really is a story about biology stripping itself down to the absolute bare essentials.
Just the minimum viable products of life.
Exactly. It is about efficiency, stealth, and survival.
So let us start with the basics. What makes a mycoplasma a mycoplasma? Like, if I am looking at a lineup of bacteria, how do I pick this one out?
Well, the defining characteristic, the thing that separates them from virtually all other bacteria is right there in the classification description.
Right.
They completely lack a cell wall.
No cell wall at all.
None. Zero. If you think about most bacteria, like E. coli or Staphylococcus.
The ones we hear about all the time.
Right. They have a rigid structure on the outside. It is made of a mesh called peptidoglycan.
Peptidoglycan, okay.
Think of it like an exoskeleton or a suit of armor.
So it holds them together?
It holds them together, it protects them from the environment, and it gives them their distinct shape.
And mycoplasmas they just skipped that part of evolution.
In a way, yes. They simply do not have it.
So they are essentially naked.
In a biological sense, yes.
Yeah.
And because they do not have that rigid wall, they are what we call pleomorphic.
Pleomorphic. Let's unpack that word for the listener.
Sure. Pleo meaning many, and morph meaning shape.
Many shapes.
Right. Because they lack that rigid outer shell, they do not have a fixed standard shape.
Like a water balloon versus a cardboard box.
That is a perfect analogy. The box has a shape, it stays a box. The water balloon takes the shape of whatever it is pressing against, or it just wobbles around.
Just a blob.
Exactly. The text actually describes them as protoplasmic masses of indefinite shape.
That sounds like a sci-fi monster. Protoplasmic masses. It sounds like something from The Blob.
It is surprisingly accurate, though. If you look at them in liquid media, they take on many different forms.
Like what?
Pears, balloons, long filaments.
Wow.
And if you grow them on solid media, they look easily disported. They possess this plasticity and delicacy that makes them incredibly difficult to study using standard bacteriologic methods.
Because you cannot just treat them like typical bacteria where you can predict exactly what they will look like under a lens.
Right. You cannot rely on morphology.
And their size, or really their lack of size, is another huge part of this, right? The source material makes a massive point about just how tiny these things are.
They are shockingly small. We are talking about a range of 125 to 250 nanometers.
Nanometers. Okay, let us try to visualize that for everyone.
It is hard to visualize. But to put that in perspective, in the lab, if we want to sterilize a liquid.
Like to remove all the bacteria.
Yes. We usually push it through a specialized filter. Those standard filters typically have a pore size of 450 nanometers.
Okay, so a 450 nanometer hole.
Right. And that catches almost everything.
Like a pasta strainer catching the noodles.
Exactly. The bacteria are too big to fit through the holes. But mycoplasmas,
they are 125 to 250.
Right. They are so small and so squishy because they lack that rigid wall that they just slide right through those 450 nanometer pores.
Wow. So if you were a scientist, say, 50 years ago trying to filter out bacteria to get a sterile liquid.
These guys might just slip right through the net and you would never even know they were there.
That is incredible.
And that extremely small size puts them in a category comparable to large viruses or chlamydia.
So they are kind of living in this weird middle ground between bacteria and viruses.
In terms of size, yes. In fact, Mycoplasma genitalium, which we will discuss later, has the smallest genome of any known mycoplasma.
Smallest genome meaning the least amount of DNA.
Exactly. Its genome is barely twice the size of some large viruses.
That is wild. It is barely more than a virus, but it is still technically a bacterium.
Yes, and that distinction is crucial. This is the hook for why they matter so much. They are the smallest organisms capable of free-living existence.
Free living, meaning they do not need a host cell to replicate.
Correct. Viruses are obligate parasites. They absolutely must hijack a host cell's machinery to make copies of themselves.
They are just code in a shell.
Exactly. But mycoplasmas, despite being tiny and stripped down, can self-replicate on laboratory media.
So they can grow on their own.
Yes, they have their own ribosomes, their own DNA replication machinery. They are life, but at the absolute minimum setting.
Life at the minimum setting. I love that phrasing. So we have these tiny, shapeless, wall-less blobs. But if they do not have a wall, what holds them together? Like, why don't they just dissolve?
Well, they do have a membrane, specifically a triple-layered unit membrane.
A unit membrane.
Yes. But here's where it gets really interesting chemically, because they do not have that tough wall to provide structural integrity on the outside.
They need something else.
They need something else to stabilize that delicate membrane from within.
And that something is cholesterol.
It is. Sterols, specifically. The text highlights that their membrane contains a sterol.
Which is wild because bacteria usually do not deal with cholesterol, do they? I thought that was strictly an animal thing.
You are right. Usually, bacteria synthesize their own wall components and do not need sterols at all.
Right.
But mycoplasmas cannot synthesize these sterols themselves. They just do not have the genetic machinery for it.
Because they have such a tiny genome.
Exactly. So they are parasitic in that sense. To grow them in a lab, the medium absolutely must contain serum or cholesterol to produce those sterols for growth.
So they are incredibly high-maintenance eaters.
Sorry.
You cannot just give them sugar water like normal bacteria. You have to give them serum. They need to steal the structural stabilizer from the host.
Precisely. They are entirely dependent on the host or the lab technician to provide that structural stability. They literally build their bodies out of the fats of their host.
That is vaguely unsettling. Speaking of eating, what about their metabolism? The text breaks down a few different types based on what they actually consume for fuel.
Right. Many mycoplasmas use glucose as a source of energy, which is fairly standard.
Sugar is fuel.
Sugar is fuel. But there is a specific group, the ureaplasmas, that have a very unique requirement.
Ureaplasma, I feel like the name kind of gives it away.
It does. Ureaplasma requires urea.
Urea, like in urine.
Yes. Specifically, the source notes they need 10% urea for growth.
10%? That seems like a lot.
It is a significant amount.
Yeah.
And they hydrolyze it.
Meaning they break it down chemically.
I do.
Yeah.
And in the lab, you can actually see this happen because breaking down that urea causes a distinct color change in the media.
Oh, that is a handy trick for scientists. So you have the sugar eaters and the urea eaters, and then there's the issue of oxygen. We usually categorize bacteria by whether they love oxygen or hate it. Where do these guys fall?
Well, it depends on the species, which adds another layer of complexity.
Of course it does.
Mycoplasma pneumonia, the lung pathogen, is an aerobe.
Meaning it likes oxygen, which makes sense. It lives in the lungs. Plenty of air there.
Exactly. It is perfectly adapted to its environment. But Mycoplasma hominis and Ureaplasma urolithicum,
which live elsewhere.
Right. They're found in the urogenital tract,
Sure.
and they prefer microaerophilic or anaerobic conditions.
Low oxygen or no oxygen?
Yes, because there is significantly less oxygen deep in the tissue or the genitourinary tract.
So they really are extreme specialists.
They absolutely are. They have stripped away everything they do not strictly need and optimized completely for their specific biological niches.
Okay, so we have the underlying biology down. They are small, wall-less, shapeless, needy for cholesterol, and adaptable to different oxygen levels depending on where they live.
That is a good summary.
Now, the source material says there are over 200 known species. We obviously are not going to cover 200 species today.
No, that would be an incredibly long deep dive.
Right. We are focusing on the ones that actually matter to us humans. The text says at least 16 species are of human origin.
But we are really drilling down on the big four that cause the vast majority of the trouble.
The big four. Let us introduce the lineup for everyone. Who is first?
First is the heavyweight, Mycoplasma pneumonia.
The lung guy.
Right. As the name suggests, it causes pneumonia, and it is also associated with joint and other infections. This is the one you are most likely to encounter in a general clinical setting.
Okay, so that is number one. Who is number two?
Mycoplasma hominis.
Hominis.
This one is strongly linked to postpartum fever and has been found alongside other bacteria in uterine tube infections.
So a major player in women's health.
Definitely.
And number three?
Ureaplasma urolithicum.
The urea eater.
Yes. This is a recognized cause of non-gonococcal urethritis in men.
Non-gonococcal meaning not gonorrhea.
Correct. And it is also associated with severe lung disease in premature infants of low birth weight.
Wow. Okay, we will definitely come back to that. And finally, the newcomer.
Mycoplasma genitalium.
Genitalium.
It is closely related genetically to Mycoplasma pneumonia, but it hangs out in the urogenital tract instead of the lungs.
What does it cause?
It is associated with urethritis and other infections, and it is becoming a much bigger topic of conversation in sexual health right now.
Okay, so that is our cast of characters, the big four. Now, I want to talk about how scientists actually find these things in the real world. You mentioned earlier that they are invisible to standard microscopes.
They are.
So how do you grow the ungrowable?
It is incredibly difficult. The text actually explicitly states that direct microscopic examination of patient specimens is useless.
Useless. That is such a strong word for a medical textbook. Usually they hedge their bets and say it has limited utility or something.
It is strong, but it is accurate. Think about the Gram stain.
Right, the Gram stain.
It is the most basic test in all of microbiology. You swab a sample, you dye it, and you stain the bacterial cell wall, either purple or pink.
Positive or negative.
Exactly. Well, these guys have no cell wall.
Oh.
So you simply cannot Gram stain them. The dye has nothing to grab onto.
So they just stay invisible.
Yes, and they are just too small to see clearly with a normal light microscope anyway. So you have to culture them.
You have to grow them in a dish.
But even that is a massive ordeal.
What does the menu look like for these bacteria? If I am a lab tech and I want to grow them in a petri dish, what exactly do I have to feed them?
You need a very, very rich medium. You cannot just use standard agar jelly.
Because they are needy.
Very needy.
Yeah.
You need serum for the cholesterol we talked about.
Right.
You need a metabolic substrate like glucose or urea, depending on the species. And you need complex growth factors like yeast extract.
A very specific recipe.
A very specific, highly enriched recipe.
And apparently you need a lot of patience, too.
A lot of patience.
Yeah.
There is a real divide here between the rapid growers and the slow growers.
Okay.
Mycoplasma hominis and Uriplasma urolithicum are relatively fast.
Yeah.
They take about 2 to 4 days to grow.
2 to 4 days is reasonable for a lab test.
It is manageable. But Mycoplasma pneumonia, you could be waiting up to 4 weeks for that culture to grow.
4 weeks. That is an entire month. If I have pneumonia right now, I do not want to wait a month to find out what is causing it.
Exactly. By the time the culture finally comes back positive, you are either completely better
or things have gone very badly.
Let's hope better.
Right.
But yes, that is a major clinical challenge, which we will get to when we talk about modern diagnosis. But when they do finally grow on the agar, they produce something very distinct.
The fried egg.
Yes. It is one of the most famous visual descriptions in all of microbiology. The fried egg appearance.
That is such a specific, weird image for bacteria. Paint a picture for us. Why does it look like breakfast?
So remember how we said they do not have a rigid structure?
Right, the water balloon.
When a normal bacterium grows on agar, it piles up on top of the surface like a little mountain or a dome.
Okay.
But mycoplasmas are fluid. The center of the colony actually embeds itself beneath the surface of the agar.
It digs in.
It digs right in. It grows down into the jelly.
Wow.
This creates a dense, dark center. But the edges of the colony grow outward on the flat surface, creating a lighter, granular periphery.
I see.
So when you look at it, usually you have to use a hand lens or a low-power microscope because the colonies are tiny, maybe 20 to 500 microns.
Just a fraction of a millimeter.
Right. It looks exactly like a microscopic fried egg. The dark yolk is in the middle where it dug down, and the lighter white is spread out on the outside.
That is so strange, but the text notes that Mycoplasma pneumonia colonies are smaller and might actually lack this specific look, right?
Yes, Mycoplasma pneumonia is always the difficult one. Its colonies are minute, and they might not show that classic fried egg morphology perfectly. It plays hard to get.
Of course it does. And what if you try to grow them in a broth instead of a solid jelly, like a liquid culture?
That is even trickier. Usually if bacteria grow in a liquid, the liquid gets cloudy.
Like it gets murky.
We call it turbidity. It is like pouring a drop of milk into a glass of water. But with mycoplasmas, because they are so incredibly small, and they do not refract light the same way normal bacteria do, there may be absolutely no turbidity even when the bacteria are present in massive numbers.
So you look at the test tube, it looks perfectly clear, and you think nothing grew.
But actually, it is full of invisible pathogens.
Millions of them.
Yes. You could have a massive infection in that tube and not see a single thing with the naked eye. You actually have to do specific Giemsa stains of the centrifuge sediment just to see those characteristic pleomorphic structures.
It seems like these organisms are perfectly evolved to be stealthy.
Stealthy is the perfect word for it. And that leads us right into their antigenic structure.
The antigens, meaning the proteins on their surface.
Yeah. The text mentions that Mycoplasma pneumonia and Mycoplasma genitalium have highly evolved systems to vary their outer membrane antigens.
Meaning they change their coat to hide from the immune system?
Precisely. They constantly change the proteins on their surface. So the host's immune system has a much harder time recognizing and targeting them.
Like a disguise.
Yes. Just when your body's antibodies learn to attack pattern A, the bacteria flips a genetic switch and changes to pattern B.
Wow.
It is a brilliant survival strategy called antigenic variation. It keeps them one step ahead of your immune defenses.
Okay, so they are incredibly small, they are stealthy, they're hard to grow, and they constantly change their appearance. But how do they actually hurt us? Let us move into Section 3, pathogenesis.
How they cause disease.
Exactly. The text mentions something called the tip.
Right. This is crucial for understanding how they work. Most bacteria that cause disease need a way to stick to the host tissue.
Because if you cannot stick, you just get washed away.
Exactly. You get washed away by mucus or urine or saliva. Now, mycoplasmas remain extracellular.
Meaning they stay outside the host cell, they do not burrow inside.
Correct. They stay on the outside attached to the surface. And to do that, many of them have evolved a specialized flask or filamentous shape with a very specific tip structure.
A flask shape, so they have a little handle or a neck.
In a way, yes. Think of it like a specialized suction cup or a grappling hook right at one end of the cell.
Okay.
And that tip is completely packed with interactive proteins called adhesins.
Adhesins, like adhesive glue.
Exactly. It is biological glue. For Mycoplasma pneumonia, the specific protein is known as the P1 adhesin.
P1.
And for Mycoplasma genitalium, it is the MGP adhesin.
The text actually gets pretty specific about the chemistry here. It says these adhesin proteins are proline-rich. Why does that matter to us?
Well, proline is a specific amino acid that heavily affects how proteins fold up into their 3D shapes.
Okay, protein folding.
Yes, it makes the protein structure very rigid and stable. Being proline-rich likely aids in the structural folding and the intense binding capability of these adhesins.
It makes the glue really strong and durable.
And makes the glue exceptionally effective.
And what exactly are they gluing themselves to?
They latch on to very specific receptors on your cells, specifically CLE glycoconjugates and sulfated glycolipids.
CLE glycoconjugates, that is quite a mouthful.
It is a mouthful. But essentially, they are targeting specific, complex sugar molecules that sit on the surface of the mucosal cells lining your respiratory tract or your genital tract.
So they find the sugar and they lock on.
Yes. And Mycoplasma pneumonia specifically targets ciliated cells in the respiratory tract.
The ciliated cells, those are the ones with the tiny little hairs that constantly move mucus out of your lungs, right?
Correct. Your lungs have this amazing continuous system called the mucociliary escalator.
They escalate.
Right. The mucus traps dust, pollutants, and bacteria. And the microscopic cilia beat in a coordinated rhythm to constantly sweep that mucus up and out of your airways.
So you can swallow it or cough it out. It is the primary defense system.
Exactly. And if you look at the electron micrograph provided in the source text, you can actually see the Mycoplasma bacteria physically attached right at the base of these delicate cilia.
Just clinging to the bottom.
Yes. And what happens is, once they attach, they inhibit the cilia.
They stop the sweeping motion.
They completely stop the sweeping. They jam the gears of the escalator.
That makes perfect sense. This explains so much about the symptoms. This is why you get that persistent, dry cough. Your body's natural mechanical system for clearing out the airway is broken.
The mucus just sits there, stagnating.
And you just have to forcefully cough to get anything out.
Exactly. And unfortunately, it gets much worse than just a jammed escalator. So they attach, they stop the cilia, then they start doing actual damage to the cells.
How do they do that? Do they release toxins? I was going to ask if it is a toxin like botulism or something.
Not a classic toxin in the traditional sense, but they do cause direct cytotoxicity.
Cell death.
Right. They generate hydrogen peroxide and superoxide radicals.
Hydrogen peroxide, literally the stuff in the brown bottle I pour on a cut to clean it.
The very same chemical. But imagine that highly reactive chemical being produced right against the delicate membrane of your lung cells.
Oh.
They are essentially producing potent oxidizing agents that chemically burn and degrade the host cell membrane directly.
They are bleaching our lung cells from the outside in.
Effectively, yes.
Yeah.
And because they are strictly parasitic, remember, they need our nutrients to survive.
Right, the cholesterol.
So there's also the factor of nutrient theft. They aggressively compete for and deplete the vital nutrients that the host cell needs to survive.
So they lock on, they starve the cell, and they burn it with peroxide at the exact same time.
It is a brutal, multifaceted combination. Physical attachment, chemical burn, and starvation.
It really is. And this brings us right to the specific diseases they cause. Let us start the big one we keep mentioning. Mycoplasma pneumonia.
The respiratory threat.
The text says this is endemic worldwide.
Everywhere.
Yeah.
It is not a tropical disease. It is not regional. It is everywhere humans are. And it has a very specific age bracket where it is most prominent.
Which is?
Persons aged 5 to 20 years.
Okay, 5 to 20. So school-age kids, teenagers, and young adults.
Yes.
We are talking dorm rooms, military barracks, crowded classrooms.
Exactly. High-density living situations with young, active people. It is transmitted directly person to person via infected respiratory secretions.
So coughing, sneezing, just talking closely to someone.
Right. And interestingly, the text notes, it behaves very much like a viral pathogen in how it spreads.
Meaning what?
It causes both endemic infections, meaning it is always just kind of simmering around in the background of the population.
Okay.
And it causes epidemic infections, where you get these sudden, massive outbreaks sweeping through a whole school or a military base all at once.
And immunity to this isn't perfect, is it? Like, I can get the flu vaccine, or if I get the measles, I am immune. But can I be immune to this?
No. You do develop some antibodies, specifically IgA antibodies, in the mucosal lining of the nose and throat.
Right.
But they do not guarantee full protection. The text specifically states that second attacks can happen.
So you can get this miserable cough more than once in your life.
You absolutely can.
Let us talk about what it actually feels like, the symptoms. The incubation period is surprisingly long, isn't it?
It is very long, 1 to 3 weeks.
Wow.
Compare that to the flu or a common cold, which usually hits you in just 2 or 3 days. This is insidious, to quote the text directly.
Insidious, slow and subtle. You do not wake up one morning feeling like you got hit by a truck. You just slowly slide into feeling terrible.
Exactly. Starts with generic malaise, maybe a low fever, headache, a sore throat. You just feel kind of off for a few days.
And then the cough starts.
Then the cough starts. And the cough changes over time.
How so?
Initially, it is non-productive. It is a dry cough. Because, as we discussed, the cilia are stopped, but there isn't a massive amount of fluid buildup yet.
Just that tickle you cannot get rid of.
Right. And it can be paroxysmal, meaning you have these sudden violent fits of coughing that you completely lose control over.
That sounds exhausting.
It is. But later in the disease course, it becomes productive. You start bringing up fluid, and you might even see blood-streaked sputum as the actual tissue damage in the lungs sets in.
And here is the classic paradox of this disease, right? The whole walking pneumonia concept.
Yes, the text describes this dissociation perfectly. The patient often looks only moderately ill.
Moderately ill, so they're walking around.
They are walking around, maybe feeling really tired, but functional. They are definitely not bedridden or gasping for air.
But inside?
But if you take an X-ray, a radiograph of their chest, you see striking consolidation.
Consolidation meaning the lungs look terrible on the film.
Yes. Cloudy, filled with fluid and inflammatory cells. The physical signs you see when you look at the patient just do not match the severe picture inside their lungs.
That dissociation is a hallmark clue for doctors, isn't it?
It is a massive red flag that points right to mycoplasma.
And usually people eventually recover from this, right?
Usually, yes. Natural recovery takes anywhere from 1 to 4 weeks.
A long time to be coughing.
It is. Death is very rare, and when it does happen, it is usually attributed to cardiac failure in people who are already vulnerable.
Okay.
And here is where the biology gets really weird. There are complications. When this bacteria decides to go off script, it goes really off script. It is not always just a lung problem.
Let us list these complications off because some of them in the text are frankly shocking.
First is hemolytic anemia. We will talk about the exact mechanism for this in the diagnosis section, but essentially, the infection can trigger your body to cause your own red blood cells to burst.
Not good at all.
Then there is bullous laryngitis.
Bullous laryngitis, which is what exactly?
Blisters on the eardrum.
Blisters on the eardrum. That sounds incredibly painful.
It is excruciating. It is a hemorrhagic, literally a blood blister forming directly on the tympanic membrane.
Oh my goodness.
And the text makes a very curious note here. It says it can occur spontaneously during an infection or in experimentally inoculated volunteers.
Wait, experimentally inoculated volunteers? Someone actually signed up to have Mycoplasma intentionally put in their ear?
Medical science has a fascinating, sometimes painful history. They likely inhaled it as part of a study, and the severe ear infection was an unexpected result.
That is what.
But yes, blood blisters on the eardrum.
And the list does not stop there. The skin.
Erythema multiforme. These are distinctive, target-shaped red inflammatory lesions that appear on the skin.
Variant issues.
Meningitis, meningoencephalitis, mono and polyneuritis. It can absolutely cross over and affect the central and peripheral nervous system.
Heart, pancreas, joints.
Myocarditis, pericarditis, pancreatitis, arthritis.
Good grief.
Because the organism can incite these systemic autoimmune-like reactions, it can affect almost any major organ system if it spreads or triggers the wrong cascade in your immune response.
So it is usually just a really annoying cough, but it can turn into a whole body nightmare.
Precisely. It is definitely not a bug to be taken lightly.
Okay, let us leave the lungs for a minute and move south. Section 5, the genital mycoplasmas.
This is a complex group to talk about clinically because unlike Mycoplasma pneumonia, where if you find it in the lungs, it is definitively a problem.
Right.
These genital mycoplasmas can sometimes just be there.
They can just be part of the normal flora, just hanging out harmlessly.
To an extent, yes. For example, Mycoplasma hominis is actually found in the oropharynx, the throat of fewer than 5% of healthy adults. But it is much more commonly found in the genital tract. And the prevalence there is strongly linked to one specific sociological factor.
Which is?
The number of lifetime sexual partners.
The lifestyle link.
Yes. The text gives us some very clear statistics. In asymptomatic men, meaning men with no symptoms of disease, Mycoplasma hominis rates are about 1 to 5%.
Pretty low.
But in asymptomatic women, it is surprisingly high, 30% to 70%.
70% of asymptomatic women. That is huge. That means the majority of women might carry this organism without ever knowing it.
It is a massive number. But here's the catch. If you change the setting and go to a sexually transmitted disease clinic, the colonization rates skyrocket.
How high?
Over 20% for men and over 90% for women.
So almost everyone walking into an STD clinic setting carries this bacteria, even if it is not the primary reason they went to the doctor.
Right, which makes proving it actually causes disease very tricky. We call it the association versus cause dilemma.
Just because it is at the scene of the crime does not mean it pulled the trigger.
Exactly. But Mycoplasma hominis is definitely a bad actor in certain specific situations.
Like postpartum fever.
Yes. It has been isolated directly from the blood of about 10% of women suffering from postpartum fever.
So it basically takes advantage of the physical trauma of childbirth to cross over and enter the bloodstream.
Yes. It is an opportunist.
And it is also linked to pelvic inflammatory disease.
Esophagitis, specifically inflammation of the fallopian tubes.
Yeah.
It is usually found mixed with other bacteria there, but it's definitely part of the infectious mix. And it is also linked to pyelonephritis, which is a kidney infection.
Okay, let us talk about Ureaplasma ureolyticum, the urea eater.
This one is interesting structurally because the text distinguishes heavily between BioVar 1 and BioVar 2.
What is the difference between a BioVar?
It is basically a substrain. BioVar 1 is now often formally called Ureaplasma parvum, and the consensus is that it is mostly harmless. Actual clinical disease is associated with BioVar 2.
And what diseases are we talking about for BioVar 2?
In men, it is a recognized cause of non-gonococcal, non-chlamydial urethritis.
So if a man has urethritis and tests negative for gonorrhea and chlamydia, this is one of the prime suspects.
Exactly.
And in women?
In women, it is very common in the genital tract, but the actual association with causing disease is considered weak.
Okay.
However, there is a very serious, well-documented association with infants.
Premature infants.
Yes. It is heavily associated with lung disease in premature, low birth weight infants.
How do they get it?
They acquire the organism directly from the birth canal during delivery.
Right.
And because their tiny lungs are so underdeveloped, it can cause severe pneumonia. It is a really tragic complication.
That is awful. Now, the text also mentions infertility. I feel like I hear about ureaplasma and infertility a lot in online forums.
The text is extremely skeptical here. It explicitly says the evidence linking ureaplasma to involuntary infertility is, at best, marginal.
At best, marginal.
Yes. So we should be very careful about jumping to major conclusions there. The internet might say one thing very loudly, but the medical textbook is highly cautious.
That is really good to know, separating fact from internet fiction. Finally, let's look at the newer culprit, Mycoplasma genitalium.
This is the one scientists are really watching closely right now. It is incredibly difficult to culture, even harder than the others. So most of our hard data mostly comes from modern molecular tests, NETs.
And it seems more definitively pathogenic than the others. Like, it is definitely causing problems.
It does seem that way. In men, it clearly causes acute and chronic non-gonococcal urethritis.
And in women?
In women, it is linked to cervicitis, endometritis, salpingitis, and yes, infertility.
And how common is this one?
About 10% of women in sexually transmitted disease clinics carry it. It is a very significant pathogen that often gets completely overlooked simply because it is so hard to find with traditional tests.
Which brings us perfectly to the next major section, diagnosis, section 6.
The detective work.
Exactly. How do we actually find these invisible, slow-growing, shapeless bugs in a real patient?
Well, as we established, direct microscopy is totally out. You can stare down that microscope all day, you will never see them.
Right.
Culture is technically the traditional gold standard, but for Mycoplasma pneumonia, waiting 4 weeks just isn't practical for treating a sick person.
So what do we do?
We look for secondary clues. One of the oldest and frankly most fascinating clues is serology.
Looking for antibodies in the blood.
Yes. And there is a very specific, weird test called cold hemagglutinins.
Okay, this is the weird science part I was waiting for. Explain this test.
So in about 50% of untreated Mycoplasma pneumonia patients, they naturally develop a specific type of antibody, usually IgM class.
Okay.
These antibodies have a bizarre property. They bind to the I antigen on human red blood cells.
They bind to our own red blood cell.
Yes, but only at cold temperatures.
So if you cool the blood down.
The red blood cells literally clump together. They agglutinate.
That is bizarre. Why does the immune system do that?
It is almost certainly a cross-reaction. The antibody the body produced to attack the mycoplasma just happens to perfectly fit the antigen structure on human group O red blood cells.
It is a case of mistaken identity.
Exactly. The immune system gets confused.
But does this clumping actually hurt the patient in real life?
It absolutely can. This is the exact mechanism behind the hemolytic anemia complication we mentioned earlier.
Oh, wow.
If the clumping happens in the cooler extremities of the body, like your fingers, your toes, or your ears, on a cold winter day, it can physically damage the red blood cells, causing them to lyse or burst open when they return to the warmer core of the body.
That is terrifying. So the laboratory test for this is essentially drawing blood and checking, does it clump up when we put it in the fridge?
Basically, yes. The antibody titer rises to a maximum at about 3 to 4 weeks into the infection.
But it is not a perfect test.
No, it is not highly specific. Other things can cause cold hemagglutinins like mononucleosis or certain blood cancers. So it is a great clue, but not a definitive answer.
So what is the definitive answer today? If I am a doctor and I want to know for sure right now.
Today, it is all about nucleic acid amplification tests, NETs, or PCR.
PCR, polymerase chain reaction, looking for the actual DNA of the bug.
Exactly, molecular detection. The text explicitly states these are the method of choice for difficult organisms like Mycoplasma pneumonia and Mycoplasma genitalium.
Because it is fast?
It is fast, it is incredibly sensitive, and you do not need to keep a petri dish warm for a month.
But there is a catch with PCR, isn't there?
There is always a catch in diagnostics. Because these molecular tests are so highly sensitive, they can easily be positive in the complete absence of clinical symptoms.
Meaning you just found a few dead DNA fragments or the person is just a carrier.
Right. You might be a carrier, you might have a few bacteria colonizing your nose, but you're not actually sick.
So a positive test plus clinical judgment is key.
Always. You treat the patient, not the lab test result. If they aren't coughing or sick, you generally do not blast them with antibiotics.
Okay, speaking of antibiotics, let us talk about treating the patient. Section 7, treatment. This is where all the strange biology we started with becomes completely crucial.
It all comes full circle right here.
The most common antibiotics people know and take are penicillins and cephalosporins. You know, amoxicillin, Keflex, Augmentin.
The beta-lactams. That is the chemical family name.
Right. And how do those traditional drugs actually work?
They work by physically attacking the bacterial cell wall synthesis. They bind to the proteins and stop the bacteria from building that rigid peptidoglycan shell.
They essentially smash the construction site so the wall cannot be built.
Exactly. The bacteria burst without its wall.
And our tiny friends, the mycoplasmas,
Have absolutely no cell wall to begin with.
so penicillin is.
Completely useless. It is exactly like trying to break a window in a house that has no windows. The chemical target simply is not there.
So if a doctor suspects walking pneumonia, but they just hand out a standard Z-Pak or amoxicillin without thinking, well, wait, amoxicillin is going to do absolutely nothing.
Correct. Giving a beta-lactam is a very common clinical pitfall. You have to actively choose drugs that work on entirely different mechanisms inside the cell.
So what does work? If we cannot break the outer wall, what machinery can we break?
You need to attack the internal factory.
Mm-hmm.
Tetracyclines and erythromycins, the macrolides, are the go-to choices.
Okay.
They physically attack protein synthesis inside the cell, specifically targeting the bacterial ribosomes.
So doxycycline, azithromycin, these are the heavy hitters for this specific bug.
Yes. And for Mycoplasma genitalium urethritis, the text specifically mentions that a single large dose of azithromycin is the typical regimen.
Why a single dose? Why not a standard week of pills?
It comes down to compliance. If you give a patient a pill they have to remember to take for 7 days, they very often stop after 3 days when they start feeling better.
I mean, which breeds resistance.
Yeah, exactly. If you give them 1 megadose right there in the clinic, you physically watch them swallow it. You know for a fact they took the full treatment.
And that stops the spread.
It drastically reduces transmission to future partners because you know the therapeutic dose is already in their system.
That is smart medicine. What about fluoroquinolones?
They are also a solid option. Drugs like levofloxacin. They attack DNA replication instead of protein synthesis. So we definitely have tools, but we have to be smart enough to pick the right ones.
But are there resistance issues? Like, are the bacteria fighting back against these two?
Unfortunately, yes. It's a constant evolutionary arms race. Some ureaplasma strains are already highly resistant to tetracycline.
Oh, man.
And Mycoplasma genitalium is becoming increasingly resistant to macrolides worldwide. It's a rapidly growing public health problem.
And the text mentions one more major limitation of our treatments. Do these drugs always permanently kill the bacteria completely?
Not always. The text notes that the drugs usually cause clinical improvement. The patient feels much better, the fever finally goes away, but they do not always completely eradicate the bacteria from the body.
Why not? If the drug kills them, why are they still there?
Because the bacteria can hide. They can go intracellularly.
Inside our own cells. I thought you said they were extracellular.
They mostly are, but they can occasionally hide inside the cells, where the circulating antibiotics have a much harder time reaching them at high enough concentrations to kill them.
Wow.
So you might completely stop the outward symptoms, but the patient could still carry the bug internally for a while.
That is deeply unsettling. They are bunkering down to survive the chemical attack. So what about prevention? Is there a vaccine we can just take?
No. There are 0 vaccines available.
None at all.
None. Many attempts have been made over the decades, but nothing has proven effective and safe yet.
So how do we stop it?
For Mycoplasma pneumonia, basic isolation is incredibly difficult because it spreads exactly like a common cold or flu, and often people spread it before they even realize they're that sick.
And for the genital types?
Standard safe sexual practices, barrier methods, condoms. Since it is strictly transmitted by direct mucosal contact, that is really the only primary defense we have.
Okay, we have covered a massive amount of ground today. We've gone all the way from the microscopic nanometer scale up to global epidemiology. Let us try to summarize the real key takeaways from this text for the listener.
I think the absolute first takeaway is the biology. The unique wall-less nature defines everything about them.
Right.
It defines their shapeless morphology, their fragility outside the body, their bizarre need to steal cholesterol, and most importantly, their complete and total resistance to penicillin.
Second is the cast of characters, the big four. You have got Mycoplasma pneumonia up in the lungs, causing the classic walking pneumonia.
And then you have got Mycoplasma hominis, Edaplasma urolithicum, and Mycoplasma genitalium down in the genital tract, causing urethritis and other complex issues.
Third is the diagnostic challenge. You cannot just use a microscope. You cannot easily culture them without waiting a month.
Right. You have to rely heavily on clinical signs, maybe look for cold agglutinins if you were going old school, or rely on modern, highly sensitive PCR testing.
And finally, treatment. No cell wall means no beta-lactams. You have to pivot and use tetracyclines or macrolides to target their internal machinery.
Precisely. It requires a total shift in clinical thinking.
So here's a final thought to really chew on. We talked about how these bacteria are essentially the minimum viable product for life.
They have the absolute smallest genomes. They stripped everything away that wasn't strictly necessary.
Right. And we talked about how Mycoplasma pneumonia can structurally mimic the tip of a cell just to attach, and how it can hide intracellularly to completely avoid our antibiotics.
It is a highly, highly specialized adaptation for survival.
If we have no vaccine at all, and they can hide inside our cells, and they very often cause mild or walking disease where people don't even go to the doctor, it makes you wonder.
Wonder what?
How many chronic unexplained respiratory issues, like that lingering cough that lasts for 2 straight months, or that sudden asthma exacerbation that just will not go away, are actually totally undiagnosed mycoplasma infections simmering in the background?
It is a very valid scientific question. The text openly acknowledges that it causes chronic disease. We might be vastly underestimating its true global impact.
And considering Mycoplasma genitalium has the smallest genome of any free-living organism we know of, does it represent the absolute bare minimum required for life? Like, if you took away just one more gene, would it cease to be a living thing?
That is the philosophical edge of microbiology. At what exact point does complex chemistry cross over and become biology? Mycoplasmas are standing right on that invisible line, staring us right in the face.
It is incredible to think about. Next time you have a cough that just will not quit, or you hear someone talking about walking pneumonia, remember the invisible, shapeless, wall-less wonders we discussed today.
And maybe ask your doctor for a macrolide, not a penicillin.
Good advice. Thanks for listening to this deep dive. We will see you next time.